Okay, so genetic information. What organelle houses our genetic information? Dan.
The nucleus.
Very good. And it is so very important because it tells each individual cell what to do. If it was a computer, it would be the operating system of the computer. It tells your brain cell to transmit signals from your brain to different parts of your body. It tells your muscle cell to contract or relax based on your movements. So it tells each individual cell what to do. We need this genetic material. This second and fourth bullet point go together. We need this genetic material because we need to be able to pass it on to our offspring, and we need to be able to create new cells. What are the two types of cell division? Jamie?
Mitosis and meiosis.
Very good. She said mitosis and meiosis. Couldn't hear you from she. Apparently my iPad is a she. And that is by creating new copies, exact copies. That is called DNA replication, and that we'll get to tomorrow. It is your last two slides on your notes, but I wanted to keep all the notes of DNA together. So we're not finishing all of those notes today. The last two slides will be finished tomorrow. We've known about DNA since 1874, so about 140 years. But why wasn't it always very clear? What made it not so clear to us about DNA? 1874 versus today, exactly. The microscope. We didn't have a microscope back then, so we weren't able to see anything. Now we're able to see, and we can actually see inside it, and a whole lot more stuff. These two experiments that we're going to cover, I'm going to first go through what the experiments were, and then we'll look at the animations. Okay? In your book, you'll have additional experiments. You actually don't need to know them. We took them out of the targets. And I also took it out of your homework guided reading, so it's not part of your guided reading. The two main experiments that you need are the Griffith Experiment and the Hershey and Chase. So if you can follow along with me and not move ahead too much, that would be wonderful. What Griffith did in 1928 is he examined different strains of strep bacteria. So Streptococcus pneumonia bacteria. He examined what's called an S version of the bacteria, and that was the lethal live version of the bacteria, and it caused pneumonia in mice. What do you think happened to the mice?
Died.
They died.
Aww.
Yeah, very sad. He also examined the R version of bacteria. The R version, when he injected into the mice, did not cause pneumonia, so the mice lived. Think about E. coli. You have E. coli in your large intestines. Is that good? Yes, that is good E. coli. It creates vitamins for you, cleans out your colon, your large intestine, so that's good E. coli. Is there also a strain of bad E. coli?
Yes.
Yeah, you hear about the outbreaks of E. coli in food. So there are many different versions, strains of bacteria, both good and bad. So he injected the bad bacteria, the S bacteria, and it killed the mice. And the fine, it didn't hurt the non-living bacteria, non-live cell bacteria, the R version, and it did not kill the mice. Then what he decided to do was he decided to heat kill. He heated up that S version so high to kill the bacteria, because the S version was the live bacteria, he killed it. We also know what heating does to proteins. What is heating, extreme heat, do to proteins? Because all proteins are enzymes, or all enzymes, sorry, all enzymes are proteins. What happens to when you heat a protein? Denatures. It kills it. It destroys it. So when he heat killed this S version bacteria, injected it into the mice, what happened to the mice? Nothing. They lived. So he destroyed the cell so much that it didn't cause anything to the mice. Then he decided, I don't know what made him decide this, but he decided to mix that heat killed version of bacteria with the R bacteria, the non-lethal version. And what he found when he injected in the mice, that it actually caused pneumonia and killed the mice. So he found that was strange. And he concluded that there was some type of material, some genetic material, that was transferred from the S version, the lethal version, to the R version, causing that R version of bacteria to become the lethal bacteria and killing the mice. At that point, he didn't know if it was proteins or DNA. We now know, based on other experiments and science, that proteins can be killed by heat. And so the genetic material must be a DNA and not the proteins. But the only conclusion that he was able to make was that there was some type of genetic material that transferred and transformed in his experiments. Okay.
Mice injected with R cells remained healthy. When Griffith examined blood samples from these mice, he found no live bacteria.
So we know that. And look, cute mouse alive.
Mice injected with S cells developed pneumonia and died. When Griffith examined blood samples from these mice, he found live S cells.
It just looks like he's sleeping.
Mice injected with S cells that were killed by exposure to high temperature remained healthy. When Griffith examined blood samples from these mice, he found no live bacteria.
And what happened when we mixed the heat killed with the R?
Mice injected with a mixture of live R cells and heat-killed S cells developed pneumonia and died. When Griffith examined blood samples from these mice, he found both live R cells and live S cells. Heat killed the S cells, but did not destroy their hereditary material. Somehow.
So this is the really key thing here. He killed the S cells, the protein part of it, didn't destroy the hereditary material, the DNA part. He didn't know that it was DNA yet or the protein yet. He just knew that somehow that material, that hereditary material from S cells was able to get transferred into the R cells, causing those R cells to become killer S bacteria cells.
That material was transferred from the dead S cells to living R cells.
Okay, that is Griffith, and you have this picture showing what happened. It's a nice kind of quick looking overall picture. I know, they're cute mice and dead mice, scarring you for life, I know. So now we know that DNA, or that genetic material got transferred. Genetic material is in our bodies, in our cells. But what is the genetic material? So at this point in history, we don't know what actually the genetic material is. Is it protein or is it DNA? And so here comes Hershey and Chase and Hershey. These two men worked with viruses. I hate that they call the viruses bacteriophages, because if you look at the term bacteriophage, you think it's a bacteria, but it's not. Anything that has a phage at the end, P-H-A-G-E, that's a virus. So if you want to just call it virus, call it a virus. So he worked with viruses, and viruses are only made of DNA and protein. So now it's really, really easy to figure out if it's the DNA or the protein, because that's the only thing that the viruses are made of. What they did was they took some viruses and made the DNA radioactive by making phosphorus in the DNA radioactive, causing that radioactive. And they took some viruses and made the protein radioactive, making the sulfur in protein radioactive. Depending on which cells became radioactive, either the ones with the viruses with the radioactive DNA or the ones with the radioactive proteins, whichever was radioactive, which cells became radioactive, that's how they would determine which was actually the genetic material inside the cells. The conclusion, I'm going to show you again the animation in a second. The conclusion was that since the bacteria that was injected with radioactive DNA was the only one that became radioactive, then we knew that DNA was actually the genetic material that was injected from the virus into the bacteria cell, and it was not the protein. So this is actually what a virus looks like. It looks cartoonish, but this is what a virus looks like. In your book, it has a picture of an actual virus. It looks like this. It looks like more on the one on the right, which is the 3D one. But it's showing you the breakdown of what it's made of. So if you see on the left, the black is the protein, and the blue is the DNA. So it's only made of protein and DNA. And what happens is the DNA in here, so you see the DNA in here, it will attach to the cell and actually go into the cell from that tail. So the DNA, we will see in a second in animation, is actually the part that gets injected, not the protein. Do you have, I think you have this one. This covers the flow of the experiment. The top half you see is talking about the radioactive DNA. So you can see that that is highlighted up there, that it's only the DNA that's radioactive. The bottom is talking about the proteins being radioactive. And at the end, they looked at the cells, and whichever cell was radioactive, they knew that's what got into the cell. And so you could see this top cell, this one was the radioactive one, and that was the radioactive DNA that was injected. This one, the radioactive protein, this cell did not become radioactive, so we know that the protein did not, on the bottom, did not inject anything in. So let's see the actual animation of it. It's kind of a, I don't want to do the narrated one, but this takes a little bit of time to go through. I'm not going to read everything. This is a virus. We know the protein coat, the DNA inside. And we have, this is what happens. This does happen to your body when you have a cold. The virus attaches to the cell and will inject something in there. And then, unfortunately, the virus replicates itself inside the cell, breaks free of that cell. And what do you think the rest of the viruses do? Go to other cells. And that's how you get sick. So now what they did was they decided to, DNA contains phosphorus, protein contains sulfur, and so what they decided to do was make the phosphorus radioactive and the sulfur radioactive. So the radioactive sulfur is the protein coat, and the radioactive phosphorus was the DNA. They then put it in a hull with a bacteria. It attached the bacteria, it cloned, it replicated itself. And then the virus did its job, injected whatever material at this point into the bacteria, and they actually put it into a blender. Why would they do that? Why would we? Not exactly. You wanted to separate the cell from the viruses. So it actually shook the viruses off, and you can see it doing that right now. And it shakes off the viruses because they only want to look at find the DNA. They only want to look at the cells. Okay? So right now we have a mixture of cells and a mixture of some liquid with viruses. Those are the mixtures. Which one do you think is going to be heavier? The cells or the liquid? The cell. The cell's going to be much heavier than the liquid. It's got all the organelles and everything. And so then they poured the blender stuff into the test tube. And the cells went to the bottom of the test tube. They're called pellets. The supernatant, which was the liquid and the viruses, that was the liquid part on the top. And then we needed to see what was radioactive. If the bottom was radioactive, on this bottom one, if the bottom was radioactive here, then we knew that the phosphorus, the DNA, was down here. Right? If the bottom of the top one was radioactive, so if the pellet of the sulfur one was radioactive, then we would know that the protein actually went into the cells. But because we only care about the cells, because the cells were radioactive with the phosphorus, that's the DNA, that's how Hershey and Chase figured out that DNA was actually injected into the cells. And then it's going to show you how it actually happens. it attaches, and the DNA, I gotta press play. And the DNA actually, did I not press play? Apparently not. The DNA actually goes from the virus through the tail into the cell. And then it makes new viruses, and then it breaks free, and will then invade another cell. And that's how you, unfortunately, get sick as well. And go to the next one.
It's not your eye.
Any questions on that?
Under your